Bolton. I am a senior biology major, and I work with Dr. Matthew Gentry in the Department of Molecular and Cellular Biochemistry. Imagine what it would be like for a child whose main concerns are making new friends at school or learning how to play a sport to receive an unexpected and devastating cancer diagnosis. This diagnosis will require at least a year of chemotherapy, surgery, and radiation, and will negatively impact their standard of living even upon the completion of the treatment. Unfortunately, this is the reality for hundreds of children each year, as Ewing sarcoma is the second most common pediatric bone cancer. Around half of patients will develop metastatic disease, and at this point, the long-term survival rate is only 20%. It is crucial that we find novel ways to improve their treatment. In Dr. Gentry's lab, we look at the role of sugars and their metabolism in cancer progression. The food that we take in is broken down into sugars called glucose, which is really important for our body's energy. In healthy cells, this glucose is stored in bundles called glycogen. However, this is relevant to my project because abnormal and excessive accumulations of glycogen have been identified in several cancer types, including Ewing sarcoma. Additionally, I confirmed this in a preliminary study where I took 16 Ewing sarcoma patient samples from the University of Kentucky. I found that there was a significant increase in glycogen abundance in the patient's tumor tissue compared to other tissues. There are multiple steps in the synthesis of glycogen from glucose, but what I really want to point your attention to is a protein called glycogen synthase, which we think of as a sort of gatekeeper for the creation of glycogen. The goal of this project was to identify how inhibiting the activity of glycogen synthase would impact the overall progression of the disease. To look at this, we utilized two compounds called A and E that are glycogen synthase inhibitors. First, I looked at the ability of these compounds to reduce glycogen in a Ewing sarcoma cell model. I plated the cells and allowed them to grow overnight. Then I gave half of the cells a control of normal cell media and the other half a 40 micromolar concentration of either compound A or E. Additionally, in a mouse model, Ewing sarcoma cells were injected underneath the skin of the mice to initiate tumor growth. Then, the mice were given twice weekly injections of either PBS, a control solution, or compound A. Following the collection of both these cells and tumor tissue, I quantified the amount of glycogen within our samples. To do this, I utilized gas chromatography mass spectrometry. This is a tool that allows us to take the disordered mixture of materials within our sample and on a microscopic level place each component into an organizing bin so that it can be individually measured. I found that compounds A and E significantly reduced the glycogen abundance in Ewing sarcoma cells compared to the cells that were not treated. Similarly, mice that received an injection of compound A had a decreasing and nearly significant difference in glycogen abundance in their tumor tissue. These data demonstrate that our compounds are able to reduce the glycogen stores that this cancer uses to grow. However, further analysis revealed that these compounds alone do not have a significant impact on the ability of the cancer cells to merely survive. Therefore, our next steps were to combine our glycogen synthase inhibitors with chemotherapies so that we could not only stop the growth of the cancer, but actually kill the cancer cells. Excitingly, a combination of compound E and a commonly used chemotherapy for Ewing sarcoma patients, doxorubicin, halted Ewing sarcoma cancer cell growth and survival at multiple time points with significant differences. Future directions in this project should include identifying the most successful combination of glycogen synthase inhibitors and chemotherapies, determining at what time point to give glycogen synthase inhibitors in the patient treatment timeline, and assessing for reaccumulation of glycogen. Overall, these data further define the relationship between glycogen and Ewing sarcoma, describe the preclinical potential of compounds A and E, and with further research will contribute to a more effective treatment regimen for these children. Thank you so much for your time, and I'm happy to take any questions.
So where are you on testing the glycogen synthase with chemotherapy drugs? So are there trials or more research coming up around that?
Thank you for your question, and it's very exciting stuff. What we're doing here with compound A and E is preclinical. However, these compounds are being characterized in glycogen storage disorders in different preclinical observations, but also clinical trials with good success. And I can't go into the specifics because they're being currently used in other labs. But what's exciting is that we are characterizing these compounds for the first time in cancer. So it's very new, but we're very excited in our next steps. Thank you.
So going back to the basic science here, and I want to make sure I understand. So it sounds like, I actually wrote it down, but I can't read my handwriting. I think what you said was that the cancer uses the glycogen accumulations for growth. So it's not just that there's a correlation that you see the glycogen accumulations when you see instances of the cancer, but rather the tumors themselves, the cancer is using the glycogen accumulation for duplication and for feeding. Is that correct?
Yes, and what's interesting about that is that we see that Ewing sarcoma does use glycogen for metastasis or invasion, and let's say from a bone in your leg to your lungs. That's like what metastasis would look like, and it does use glycogen. But it's interesting that there are some cancer types that have increased glycogen that don't use it for metastasis, such as renal cancer, and we don't exactly know why. We have known for a while something called the Warburg effect, where we know that cancer cells have an altered metabolism. We know that they're relying more on anaerobic or metabolism without oxygen. We know that we're relying more on those pathways, but we're still working out why and what they're using these different fuels for. Thank you.
This is more curiosity than anything. In your methods and results, you're showing that for the plated cells, you did compound E and compound A, but then in the mouse, you don't show compound E.
Yes.
Is there a reason for that?
Because we haven't been able to do it yet, this was our first time using the mice for compound A. This was based on a preliminary study where we used a naturally occurring glycogen synthase inhibitor called guaiacol, where we not only saw reduced glycogen, but we actually measured tumor volume throughout the study and saw that guaiacol significantly reduced tumor volume as well. We couldn't do that here because the mice that we were using had some fur and it would be difficult to measure with a little ruler. But we hope to do that with compound E as well. And then with our assay looking at growth, we would like to see, what is the effect of doxorubicin by itself, compound E by itself, different mixtures, and we even want to use different chemotherapies like irinotecan and vincristine, because those are also commonly used chemotherapies for Ewing sarcoma patients. So all in a nutshell, we have so many exciting plans. We're just not there yet.
Is there also the plan then to use compound A with the chemotherapy treatments?
Yes, we would hope to do that and even bring in different glycogen synthase inhibitors, maybe try what they would call in the clinic cocktails of chemotherapies. These kids usually get 2 or 3 chemotherapies at a time.
Thanks.
Thank you. Thank you.